apoplastic and symplastic different routes of how stuff can get into a plant. And remember that our theme for this whole unit is detecting and responding to your environment and using that in order to maintain dynamic homeostasis. Remember, that's our whole theme, and we talked about that with regard to plants last class, and we're talking about that with regard to animals for the several classes prior to that. Well, one of the most difficult things for a plant to manage is water. Okay, they have to have water, just like you and I have to have water. And if you're a plant that doesn't grow on someone's manicured lawn, you're kind of subject to the wills of Mother Nature. When is it raining and when is it not? And that's a tricky thing for a plant to deal with. And so they have to have ways of conserving water when they need to, and of taking water in when it's available. And so for today, we're going to talk about transpiration, and this is the lab we're actually working on right now. Even though we didn't talk about it last class, we set it up and we said, okay, how much mass do you have right now, and how much mass is going to be moving through these plants over the course of the week? And the reality is, I was just looking at some of your data, some of you lost as much as 30 grams over the course of 2 days. And so just think, look at this, this fits in very nicely with my first data point up here. An average sized tree is transporting nearly 800 liters of water on a warm, sunny day. Imagine your little bitty plant. Some of them lost as much as 30 grams. How much is that in volume? 30 milliliters. Well, how much is 30 milliliters? Like, it's a kind of a lot in a graduated cylinder, right? I mean, that normal big graduated cylinder you use is a 50 milliliter graduated cylinder. So, 3/5 of that is kind of, or I guess not quite, because that would have been 3/10, but you see what I'm saying. A large portion of that graduated cylinder was lost through your little bitty pansy plant that's like 5 inches tall. So now if you consider that on the scale of the trees outside, the amount of water that is literally being moved out of our planet and into our atmosphere by plants is a huge quantity. It's a huge quantity. And so water, we know, starts in the soil. And we said last class that the movement of water is a one-way road. It only moves up. We'll talk about that first. Okay, we know that water is absorbed by the roots. It's actually absorbed by these modified root cells called root hairs. So here's a little bitty fuzzy one that easy to see. Each of these root hairs is actually a cell, okay, and that is responsible for most of the absorption of water and minerals from the soil. So the water goes in through those root hairs, and then it only can move up, moves up, moves up, moves up, and then evaporates out of the leaves as water vapor. Okay, and then water vapor, of course, that's part of your water cycle. Water vapor becomes the humidity in the air and the clouds that you see until there's enough water vapor that accumulates, and then it falls as rain, and then, as precipitation, rain or snow or sleet or hail or any of those things, falls as precipitation and is returned to the earth. And so it's this massive cycle, the water cycle, that plants play a huge role in through this process of transpiration. Now, of course, there's also evaporation just off the tops of lakes and the ocean and stuff like that, that's also moving water back up into our atmosphere. But plants also play a huge role in this. We have to talk a little bit about water potential. So even though soil solution itself usually has a pretty low mineral concentration, active transport allows those roots to accumulate the minerals that it needs. How do we accumulate minerals we need? We eat. Well, plants can't do that, right? I guess aside from those few carnivorous ones, plants have to get the minerals that they need from the soil solution that they're in. So they use active transport to accumulate those specific minerals that they need. And then that transport itself is short distance into the cells, and then long distance transport through the xylem up and throughout the plant. Ultimately, that soil solution goes into bulk transport in those xylem elements. So of particular importance in this process is the endodermis. The endodermis is the skin. Okay, you can think of it as like a deep layer of skin that contains something called the Casparian strip. To me, that sounds like it should be the Caspian Sea, but it's not. It's the Casparian strip. And the Casparian strip is just a very, very waxy cuticle that's inside the root itself that is actually going to force all of the things that are absorbed by those root hairs to be screened by the selectively permeable membrane. okay? Because remember we talked last class that we had both the apoplastic and symplastic routes into and through these plants. Well, the trick was our apoplastic route here was never screened by our cell membrane. And the cell membrane is the part of the plant and the part of the cell that's selectively permeable. That's going to say, okay, well, I landed in an environment as a seed. This is where I landed and this is where I grew. But, you know, there's way too much of, I don't know, we won't say nitrogen, potassium, phosphorus, ions in this particular part of dirt. I don't want to take all of that ion up. I only want a little bit of it. And so the cell membrane is the thing that's going to control what's actually allowed into those plants. And so again, that little bitty root hair, so here it is in a cartoon form. Here it's one of these, and we looked at right here. Okay. That root hair starts by providing an awful lot of surface area to volume ratio to absorb these fluids. But it could be through the apoplastic route or the symplastic route. And the trick is if it's through the apoplastic route, at some point we need these fluids to have to cross the cell membrane before we let them in our xylem elements. Because this is your bulk flow, okay? This is short distance transport, this is long distance transport. And this right here is our Casparian strip. And it's just a waxy cuticle that says, okay, all of you solutions that have not actually crossed the cell membrane because you've been engaging in that apoplastic route, you have now reached a barrier. Now you must cross a cell membrane. I get the choice. I have the opportunity as the plant to say, you know what, I've got enough of you, you're not allowed in. Okay? And that Casparian strip is the thing that prevents just letting anything from coming in. And then on the inside of that Casparian strip is where we get to our vessel elements, and that's where we have that bulk flow that is moving as a response to pressure. Once that solution got into the xylem, we call it the xylem sap, and moving the xylem sap involves the loss of water by transpiration. And that loss of water happens from leaves and other aerial parts of that plant. And unless that water that's been transpired, that part that got back out into the atmosphere in terms of water vapor, is replaced by water from the dirt, the leaves will wilt and the plant could eventually die. So there's two ways that water is moved upward in the plant. This is how, I'm going to say 99% of the water moves. This is how maybe 1% of the water moves. So I want to make this very clear. Root pressure is a very small contributor to this process. Root pressure is the osmotic pressure literally pushing water up the xylem. Why? Because remember that water is going to move from high to low solutes concentrate, or I guess water concentration. And so what ends up happening is, because the plants have accumulated through active transport the minerals that they need in their roots, oftentimes the water will follow. So do you remember when we were first learning about osmosis and I showed you guys that YouTube thing and the levels of the water were different? Okay, and I said this is water pressure, that the water will move to even out the concentration, even though it seems like it's a magic trick because gravity says they should be even. So that's what's happening with the plants. The water is going to move into the roots because the plants have actually actively transported so many minerals into the roots. The water is going to passively follow. And once the water passively follows, it will actually push up in the plant. Now, this is only going to be effective for giving water to the entire plant when transpiration itself is very, very low and soil moisture is very, very high, such as at night. I would gather that this probably never, ever, ever happens in the state of Colorado because we almost never have very high soil moisture. Unless it's in the winter, in which case we're not doing transpiration at all, right? But in the summer, we just don't have that here, okay? Cohesion tension hypothesis, that's exploiting the properties of water, and that is how the great majority of water actually moves through a plant. What it tells us is that transpiration that's occurring via evaporative water loss on the leaves is actually pulling on all of the nearby water molecules that are in the plant. And so the cohesion of water molecules, cohesion means those water molecules are sticking together. Cohesion literally transmits this pull along the entire length of that xylem all the way down to the roots. And so the xylem sap is actually under negative pressure. So the evaporation of water off of the leaves is literally pulling up the water molecules all the way from through the plants and all the way down into the roots. The transpiration is happening in the leaves of plants and begins this process of water vapor in the air spaces of a leaf that diffuse down its water potential. This is the diagram that goes with it. We're going to watch a bioflix on this in a few minutes, and it's going to show you each water molecule literally evaporating up. But in essence, what I want you to see here is that in the mesophyll layer of a leaf, do you guys remember when we did the photosynthesis lab, you had to try to get the spinach cells to sink? And how you had to like put your finger on the top and suck it down and try to get them to sink? Remember how hard that was? It was hard because normally that mesophyll layer is full of air. And so what we had to do was suck the air out of it and replace it with the CO2 water, right? That's how we were able to make that lab happen. So what this is showing you is that in all of these air spaces, each of these little blue dots is actually a molecule of water that has evaporated into the airspace from our xylem. And then here's your stomata. Remember, the stomata is that pore, that opening that connects the inside of the leaf and the outside of the leaf. The stomata was a super important structure for photosynthesis because remember, photosynthesis had to allow the CO2 to come into the leaf and the oxygen to leave the leaf. That was really important because that's what photosynthesis required. The trick is, because there's a hole, kind of like a pore on your face, because there's a hole right here into the leaf, we're also losing lots and lots of water. The goal is not to lose water. We need to keep our water, but water is lost because that pore is open. The three properties of water that are required for transpiration to occur like this are adhesion, where the molecules are sticking to the sides of the vessels, cohesion, where they're sticking to each other, and then surface tension. And the surface tension is just indicating that as the water evaporates from here, it's literally going to pull water molecules off of the surface of the water that is surrounding the cells within the leaf. So this is what we end up with. The transpirational pull on that xylem sap is transmitted all the way from the leaves up at the top to the root tips and even all the way down into the soil solution. This seems like it should be defying gravity, and in a certain extent it is. But remember, water is always going to move from high to low water potential, and pure water has a solute potential of 0. So when we look at the soil that's going to have a lot of water in it, as well as a few solutes, we're going to have a water potential in megapascals of -0.3. But some of that water moves into the plant because the plant has actually got a lower water potential. Why do we have a lower water potential? Because we have actively transported ions into our roots. Then, because your outside air up here has even a lower water potential, it is also going to pull on all of this water. And we're going to remember that water potential is a combination of two things: solute potential, but also pressure potential. And so because up here, this relative negative pressure, this pulling that's occurring from evaporation, our total water potential up here is going to be so negative that water is literally going to be pulled up the tree. This requires 0 ATP. Okay? What is this being fueled by? Sunlight. So sunlight, in addition to being the energy for photosynthesis, is that anabolic or catabolic? Photosynthesis, building bigger molecules? Anabolic, good. Okay, so in addition to providing the energy we need for that anabolic process, it is also pulling water all the way up the plant against the forces of gravity. Pretty slick stuff. So, how strong are these forces? Well, water's cohesive force within the xylem gives it a tensile strength equal to that of a steel wire. Tensile strength is, if you think about thread, how much can you pull it before it breaks? That's what tensile strength is. So like if you've ever been fishing, you can buy fishing lines that they'll say 5-pound weight test, 10-pound weight test, 15-pound weight test, right? That's just saying how big of a fish can yank on this before the fishing line's gonna snap, right? So that's what tensile strength is saying. So these properties are so strong that water's force gives xylem a tensile strength equal to that of a steel wire of similar diameter. Pretty strong stuff for just some chemical bonds. The tension on those vessel elements is created by transpiration is enough to literally stretch a tree upwards. Now, it doesn't get taller, it just gets thinner. And you can actually measure this in a tree. During the day, how big around is the diameter? At night, how big around is the diameter? And it turns out on hot days, the tree will be skinnier. Okay? We're not talking about inches, we're talking about millimeters, but nonetheless, the tree will be skinnier. And so it's kind of like if you stand still and you suck in, right? I can't even breathe, right? I'm not getting taller when I do that, right? My middle just gets more pulled in. That's what's happening to a tree too, okay? We can try that in the back. Oh, we need to go. So, and again, the plant expends no energy in order for this to happen. The process is driven by evaporative water loss, which of course is driven by the sun. So let's go ahead and watch this quick, like a 5-minute video. We're going to watch our bioflicks.
Trees must transport water and minerals to their highest leaves. How does this happen? Interestingly, trees move water by losing water. Evaporation of water from plants is called transpiration. Water diffuses from inside a leaf to the outside air through pores. Inside a leaf are air spaces. Water evaporates constantly from the surfaces of surrounding cells. To better see the water, let's show it in blue. Notice that water forms a film on cell walls, including the walls of cells in leaf veins. Within the veins are hollow, tube-like cells, shown in brown. These cells, also found in stems and roots, are part of a tissue called xylem. Water and minerals are transported from roots to veins through the xylem, and then from veins into the walls of other leaf cells. What maintains this flow? Let's look at the surface of a plant cell. A thin layer of water covers the cell wall. As water evaporates, the cell wall begins to dry out. Look at how water sticks to the cell wall. This is called adhesion. Water molecules also stick to each other. This is called cohesion. Cohesion and adhesion are the results of hydrogen bonds, the dashed lines. Because of cohesion and adhesion, transpiration creates a tension from leaf cells through the xylem to the roots that pulls water molecules up the plant. Now let's look at how water and minerals enter through the roots. Roots are highly branched, creating a lot of area for absorption. Root hairs near each root tip increase the surface area. Water and minerals, shown in yellow, can follow two paths from cell to cell within a root. In one path, they travel in the cytoplasm, which is connected between cells. In the other path, they travel within cell walls. In the path within cell walls, however, they encounter a waxy barrier that blocks entry to the xylem. To continue, the water and minerals must cross a plasma membrane. In this way, only certain minerals can enter the xylem. After moving into the xylem, water and minerals are pulled up to the leaves. Water evaporates from leaf cells into the air spaces inside leaves and exits through pores. And that's how water can reach the highest leaves of a tree.
Alyssa, are you grossed out by this?
Yeah.
I hate
the stomatas are so weird.
We don't like the stomatas?
No.
Well, you may need to sit out this next thing that's all about
Oh, God!
If you'd like to sit out, you're welcome
I'll just
to.
listen.
Why are they gross?
I'll just
I
listen.
don't know. There's like
They look
little
like-
skin
the
holes.
fear of holes.
Yeah.
Well, they are pores, just like in your face. They don't
I
get
have them on my
blackheads.
thing.
There's no zits there.
It says she doesn't like holes?
She doesn't like the stomata. She can't look at them. I don't know. I got nothing. I got nothing.
Phobia.
Another phobia.
Stomachophobia.
Clearly.
You just said.
All right, let's talk about them. If you're so disturbed you need to step out, you are welcome to. Just don't step on in front of my video. So here's the deal. Again, we have to bring ourselves back to dynamic equilibrium. A plant can't produce rainfall if there's none, right? And so they have to be able to conserve water when there isn't much. And so the rate of transpiration is regulated by this stomata. And so leaves themselves generally have really high surface area to volume ratios. And that makes a lot of sense because that means they have enough surface area to absorb light for photosynthesis, which of course it needs, and also enough stomata to bring in the CO2 for photosynthesis and release the oxygen that is the result of photosynthesis. And all of this is very, very good. The problem is this also increases the water loss that happens by the stomata. And so this is just one of my leaf impressions from that experiment or that lab we did back in the fall, where we calculated the concentration. And you might remember that we only found stomata on the bottom of our leaves. We didn't find them on top at all. And the reason is simply because you don't necessarily want the stomata on top where the sunlight is actually causing evaporation to happen even faster. And so they're all on the bottom. And so a plant's water requirement is the consequence of the system's need for ample gas exchange for photosynthesis. So this is a compromise. The plant needs to have the stomata open for gas exchange, but doesn't want to leave the stomata open for more time than they have to be because of this extra water loss. And so these two cells on either side are called guard cells, and they're the things that actually help balance this conflict between the needs of the cell to keep water as well as undergo photosynthesis. Turns out about 95% of the water a plant loses actually escapes through the stomata. Any place there's not a stomata, we're covered by a waxy cuticle that surrounds the leaf that limits water loss through other areas. The guard cells that I pointed out to you on the previous slide, these are just the cartoon versions of them. They are on either side of this hole right here that goes, this hole goes into the air spaces of the mesophyll. And they can control how open or closed that stomata is simply by changing their shape. And so there's two guard cells adjacent to each stomata, one on each side. And so the shape change of those guard cells is what changes the width of that gap for the stomata. The density of stomata, so how many of them there are per centimeter squared, is both under genetic and environmental control. Desert plants genetically have lower stomatal densities than do marsh plants. That makes sense. Desert plants are very hot. Marsh plants grow literally in water. There's no shortage of water there, so there's not a difficult compromise to be met. But this is also something we call developmentally plastic. And what that means is that the environmental conditions that a plant is exposed to will also dictate the amount of stomata that develop throughout the young years or the young months or the young days of that plant's life. So for example, a lot of light exposure and low CO2 levels during development is going to lead to increased density of stomata. Well, that makes sense because if we don't have much CO2, you're going to want more stomata to get all the CO2 into the leaf that you can, because remember, we need to be able to photosynthesize, and CO2 is a reactant for that. So how do the guard cells work? Well, when the guard cells absorb water from neighboring cells, they become more turgid, and the cells themselves are fixed at either end. And so because they're fixed at the two ends, as water gets added in and they become more and more turgid, the cells bow outwards, increasing that hole in between. When the cells lose water and become flaccid, they become less bowed and that pore closes. So that's what we end up seeing here. It's the same diagram, only now we're looking at the movement of water. And so when water comes in, we're fixed at the two ends, they bow out. When the water leaves, you can see our central vacuole is smaller here than it is here. When the water leaves, we meet here in the middle, and our stomata at that point is closed. Well, how do you get the water to move? Well, water moves passively through those protein channels we call aquaporins. Well, if water is moving passively, we have to give it something to follow, right? It's not going to just go against its concentration gradient. That's not what water does. So what happens is the guard cells will accumulate through active transport potassium ions from neighboring cells. So potassium is high in these outside cells, and then as the potassium is actively transported into the cells, and we'll use ATP to power that process, what's the water going to do? The water is going to follow it, because that's what water does. Then when we want to close our stomata, we open up the channels. We open up our potassium channels. The potassium passively goes down its concentration gradient, and of course the water then also passively follows. And so the water is only moving by osmosis, but the potassium is actually moving through active transport when it's moved into those guard cells to open them. What actually controls this? So we said the potassium is what's controlling whether the water comes or goes, but what's actually controlling the potassium? Well, in general, stomata are open during the day and closed at night. That makes sense because for most plants, we're not talking about our C4 or our CAM plants, but for most plants, having your stomata is open is crucial for photosynthesis. And so photosynthesis is only happening during the day, so we would only want them open during the day. So this prevents, by having your stomata only open during the day, it prevents the plant from losing water under conditions where photosynthesis couldn't occur. There are 3 cues that can contribute to the stomatal opening at dawn. First, it's just light. There are light receptors in the plasma membrane of these guard cells, and activating those receptors cause the guard cells to begin to accumulate the the potassium, and then of course the water follows. A loss of CO2 in the leaf is another result. Why do we lose CO2 in the leaf? Because it's all being fixed through photosynthesis. So if we have a loss in our CO2, then the stomata will progressively open if sufficient water is supplied to the leaf. That makes sense also. And then I gotta be honest with you, plants also have a circadian rhythm. They know, no brain, but they know when they should be open and closed. And so even absent both of these things, they will begin to open during the daytime when they should. Okay, similar to how later on in life, you will just wake up in the morning even if you don't set an alarm. I know you're not there yet, but at some point you will. Okay. Drought stress can also cause stomata to close because even if we have plenty of light, we're short on CO2, and it's daytime, if it is really drought, we have a lot of dry days, we probably don't want our stomata open. And so a hormone called abscisic acid is produced in the roots and the leaves in response to water deficiency that actually signals those guard cells to close the stomata. That response reduces the wilting, but then of course also restricts CO2. So it's certainly a trade-off because you're not going to have as much photosynthesis. And again, we just need to keep in mind, why do the plants need water? Well, they need water to keep their stomata open, which allows them to take up the CO2. But remember that water is a substrate in photosynthesis. Plants need water in order to photosynthesize. If you go back and you look at that equation from Unit 3, water is a reactant, so we've got to have it. But don't forget that plants also need oxygen. We don't often think about this because oxygen is a waste product of photosynthesis. But in essence, they need oxygen because they have mitochondria, and the mitochondria are necessary to produce ATPs from the sugars that they produce in photosynthesis. So even though oxygen is a waste product of photosynthesis, it's still a reactant in cellular respiration. So we can't pretend like the plants don't also need to use oxygen. So of course, there are some plants that are adapted to drought-like conditions. We call those plants xerophytes. If you've ever heard of somebody xeriscaping their lawn, it's when they plant particularly native plants that don't require watering. Okay, it's called xeriscaping. So these are plants that are adapted to these drought-like conditions. They might have very small leaves, carry out photosynthesis in their stems, such as cacti. They might store water in fleshy stems. Cacti or succulents are examples of those. They could be a CAM plant where they take in CO2 at night and keep their stomata closed during the day. Their stomata could be recessed in cavities called crypts to protect them from wind. They could have long white bristles to reflect intense sunlight to keep the plant itself cooler. Or they could actually be leafless and dormant for most of the year and really only produce leaves when there is sufficient rain to make it worth their while. And so these are just the diagrams that go with that. This is the same plant, and for 11 and a half months out of the year, it looks like that. And then after the one annual desert rainfall, it leaves out, and it goes through leafing and flowering and producing seeds in a very, very short period of time. And those are spread, and then it goes back to looking like this very quickly. These are those white bristles to reflect sunlight. And here's a recessed stomata. The stomata itself is stuck inside this crypt that prevents wind from literally ripping the water away from the stomata. Last slide, just to give you a little bit of vocabulary that's a little bit odd. Sources and sinks. Okay, a source is where sugar comes from in a plant, and a sink is where it goes to. So plants are producing sugars at these sugar sources. Those are typically leaves, okay, the place where photosynthesis is happening. And plants move those sugars to sugar sinks. Those are going to be the consumers or depository of those sugars. And for us, that could be roots, buds, stems, fruits. All of those things are sugar sinks. Very simple vocab, but if the first time you see it is on May 8th for your test, that could be a little alarming. So sources is where it's coming from, sink is where it's going. Okay? Good job, guys. We have 1 minute to spare on today's